Good morning, everyone. In this video, we are going to see class 11, chapter number 20, locomotion and movements. I hope you are aware of your new syllabus. In this chapter, we have to study skeletal muscle, contractile protein and muscle contraction. That is, we have to study about skeletal muscle. We have to study about contractile protein, this contractile protein and mechanism of muscle contraction. That means till here. Let us start. Movement is defined as a visible change of position exhibited either by whole organism or any part of the body. It is one of the most important characteristic of living organism. The movement of living things are autonomic, while the movement of non-living object is induced. Both animal and plant exhibit wide kind of range of movement. The movement of animals can be muscular or non-muscular. Plant for cellular and often organ movement. Let us talk about the types of movement. Amoeboid movement or pseudopodia movement with the help of finger-shaped protoplasmic extension is called as amoeboid movement. Ciliary movement in the cell or organ with the help of cilia is called as ciliary movement. Cilia is a hair-like structure, we all of us know. Flagellar movement. The type of movement occurs in protozoans like Euglena along with the sponges. In sponges, flagella of maintain the regular current of water through the canal system. Then muscular movement. Movement with the help of muscle is called as muscular movements. Let us talk about muscles. These are made up of highly specialized thin and elongated cells called muscle fiber. Muscle arises from embryonic mesoderm. It makes the 40 to 50% of human body weight. If we talk about properties, muscles show excitability, muscles show contractibility. Muscle also possesses property of extensibility and elasticity. Now let us talk about types of muscle. It is given over here only. Muscles can be skeletal muscle. It can be smooth or visceral muscle. It can be cardiac muscle. Let us talk about this in short. The muscles, skeletal muscle, the muscle function is associated with the skeleton of organism. Under the microscope, they show stripped appearance and hence are called as striated. They are also known as voluntary as their activities are under the voluntary control of central nervous system. Visceral or smooth muscle. They are found in the inner walls of visceral organs of the body like alimentary canal, reproductive tract. They do not exhibit any striations and are smooth in appearance and hence they are called as smooth muscle. Cardiac muscle, it occurs in the wall of heart and in wall of big veins where these veins enters the heart. These are striated and involuntary in nature. Presence of oblique bands and interconnected discs are characteristic features. Let us talk about detail about the skeletal muscle. This is the diagram. Skeletal muscle is made up of number of muscle bundles or fascicles held together by fascia. Each muscle bundle contains a number of muscle fibers. You can see in the diagram. Muscle fibers bonded by sacroligma and contain well-developed endoplasmic reticulum specialized for calcium storage in sacroplasm. Muscle fibers is a cytentium and its sacroplasm contain many nuclei. There are a large number of parallel arranged of filaments called myofilaments or myofibrils. Let us talk about detail of myofibrils. A myofibril has alternate dark and light band. You can see over here myofibrils. Dark and light band. The dark bands are called anisotropic band or A band, which contain myosin. Myosin is the name of the protein. The light band are called isotropic band or I band, which contain protein actin. The striated appearance of fibril is due to the distribution of pattern of protein, actin and myosin in the muscle tissue. Both these proteins are arranged as a rod-like structure parallel to each other and also to the longitudinal axis of myofibril. Actin filaments are thinner than myosin filaments. Each I band has its central dark band called Z-line. It is also called as Z-line or Z-line membrane. This the part of myofibril between the two successive Z-line is called a sarcomere. A sarcomere consists of A band and half of its adjacent I band. A thin fibrous membrane called M line present in the middle of A holds the thick filament together. The A and I are arranged alternatively throughout the length of myofibril. Let us talk about the structure of protein. These are the structure of protein. The structure of contractile protein, that is a thick myofilament, are formed by myosin protein. The thin myofilament are formed by three types of protein called actin, tropomyosin and troponin. These four proteins are collectively called contractile protein. Thick myofilament or primary myofilament. Each myosin filament is a polarized protein made up of many monomeric proteins called meromyosin. Each meromyosin has two important pairs, which are globular head and tail. You can see in the diagram. Head, it is a short arm called heavy meromyosin, HMM. The HMM component projects outward at a regular distance and angle from each other. From the surface of polymerized myosin filament are known as cross-arm. The globular head is an active ATPase enzyme which has binding site of ATP and active sites of actin. Tail is light. You can see over here. Tail is the light meromyosin. The myosin molecules has two identical heavy chains and four light chains. The two heavy chains wrap spirally around each other to form double helix. The light chains are part of myosin head and help control the function of head during the contraction of muscles. Now let us talk about mechanism of muscle contraction. The contraction of muscle is the best explained by sliding filament theory, which states that the contraction of muscle takes place by sliding of thin and thick filament with the help of cross bridge. It reduces the length of sarcomere. This theory was proposed by Huxley, Jean Manson in England in 1954. The sequence of events leading to the contraction is initiated by the signaling central nervous system, either from the brain or from spinal cord. A motor neuron along with the muscle fiber connected to form it forms a motor unit and the action potential is conveyed to motor end plates at neuromuscular junction. A neurotransmitter is released at the junction by the neural signal which generates an action potential in sarcolemma. This spreads and causes the release of calcium ion into sarcoplasm. Calcium plays a regulatory role in muscle contraction. Increase in calcium ion level lead to binding of calcium ions, the troponin subunit on actin filament. This removes the masking of active sites from myosin. Formation of cross bridge, you can see. An ATP molecule joins the active sites of myosin head of myosin myofilament. This head contains an enzyme myosin ATPase that along with calcium ion and magnesium ion catalyzes the breakdown of ATP. The energy is transferred to myosin head which energized and straightened to join an active site on active myofilament forming cross bridge. The energized cross bridge move causing the attached active filament to move towards the central A band. The Z line is also pulled inward, causing straightening of sarcomere. The myosin head releases ADP and inorganic phosphate relaxes to its lower energy state. In repeating cycle, free head leaves the new ATP. The cycle of cross bridge formation and breakage is repeated, causing further sliding. In the next video, we are going to study our next chapter. Thank you.